Well, good evening, everyone. An unusual subject to talk about, which many people will have heard a little bit about, but this is freshwater sponges and their dependent fauna. This is the things that live within sponges. Now, freshwater sponges are, to many people, fairly obscure. Not many people know anything about them, and I've been interested in them for years, and wrote a review of the current knowledge a couple of years ago for British Wildlife magazine, which is partly where this webinar this evening has come from. There has been a bit of recent work in the UK and in Ireland, and indeed Liverpool's very own Karen Evans is one of our participants tonight, so she may say a few words later on freshwater sponges because she did her PhD on them a few years ago. So, and I will be referring to some of her work. But without any more ado, I shall move on with a general introduction to sponges and the issues to do with their identification and indeed finding them. Here we go. I've said most of this already, really. Freshwater sponges, an introduction, highlighting the difficulties of identification and also talking about the associated species, particularly the sponge flies, which are a whole family of lacewings that do eat nothing but sponges, and some sponge-eating caddisflies. As I have said to anyone who talks to me about sponges, I may come across as someone who knows a lot, but I don't regard myself as an expert because there's a lot to learn about sponges, and what I really want to do is try and get more experts out there. I'm trying to encourage more work on sponges, and I hope tonight will help do a bit of that. Sponges are actually, freshwater sponges are surprisingly common, but people tend not to even know they exist. They think of marine sponges and don't think that they're in freshwater at all. But we do have 6 species in Britain. There are 250 or so of them worldwide. And the ones we have in Britain are surprisingly common when you look for them. They are colonial structures. They're sponges, like all sponges, they are single cells that live in a colony. And therefore, they are strange for invertebrates. But I always argue that en masse, a large sponge, even a large freshwater sponge, is probably easily our biggest freshwater invertebrate, and indeed terrestrial invertebrate. So it is always surprising that they are so overlooked. And because they're so big, sometimes it's not surprising they're home to many other invertebrates. And coming back to what I was saying just now about recent work in Britain and Ireland, until recently, there was very little work on them until Karen Evans' PhD a few years ago in Liverpool, and Letitia Cogiglia and John Lucy in Ireland did a lot of work on them a few years ago too. So there are these 2 studies which help set the scene. We just need to do a lot more work to find out more about that scene. This one is a small sponge, relatively small. It's a fingered version. Some of them are literally the size of fingers, and they stick out of structures underwater and are quite obvious and quite mysterious. They are almost disturbing, really. This one is coming out of the floor of the Montgomery Canal in Wales. It's a silted up environment, but they grow on hard structures. So there's something hard under there that is silted up since it started growing, and it's pale. It's a creamy grey color. Often these big sponges are bright green because they have algae living within them in a symbiotic way. So that's quite an interesting pale version of a fingered sponge. This one is also on a canal. This is the Huddersfield Narrow. This is way back in the 1990s. I took this picture. This is a big beast. This one is probably half a meter wide and getting on for half a meter deep. When they get big, they get big. This one is green due to the symbiotic algae. And these forms are probably although we'll come on to identification later, Spongula lacustris, which is known as the pond sponge, common name, but don't be confused by that because pond sponges occur in rivers. Similarly, river sponges occur in ponds. So the common names are fairly meaningless. This is another, almost certainly Spongula lacustris, although it hasn't been sampled properly. This was on the bed of the River Thames, not in the 1990s, but last weekend. This one is definitely a pond sponge. It's not in a spot, not in a pond, and you can tell by the green. There's a slight hint of a green color. So this does have thriving algae within it. So these are fingered versions, but they're not all sponges are that spectacular. To be honest, those are just the kings of the sponges, if you like. Most of them are just like underwater lichens. They encrust structures underwater, and they grow outward slowly, and they're thicker than lichens, but they look surprisingly like lichens if you drain the water down, as in this picture here, which is the Forth and Clyde Canal in Scotland. And we're looking at a sponge growing on the walls. And this is probably a species called Ephidacia moellii, or it could be the very similar Ephidacia fluviatilis, which is known as the river sponge. But here it's not growing in a river, it's growing in a canal. So ignore the common names. Not all sponges are even that defined. This is also taken a week or so ago, this one. This is showing the edge of a river and some rocks at the edge, which have been exposed by low water. And the green on here, which looks just like a moss, isn't a moss. It's a green sponge. It's not growing like a lichen. It's growing more like a moss, but it isn't a moss. It's far too smooth to be a moss when you get your eye in. If you look right into that, you can see it's got pores rather than leaves. And that would normally be just underwater, but this dry summer has exposed it. So they are quite indefinable sometimes. This could even be like those branched ones we were looking at earlier. It could be another Spongula lacustris that's just growing in this form. That doesn't have to have fingers. There are also several other possible species. We'll come on to those. So what actually are they? It's worth working out what they are to understand them. They are definitely animals, although right up into the 19th century, people were trying to convince learned societies that they were plants. Not surprising, because a lot of them are green and branched. This picture is actually, curiously, a Thames river specimen that was described in 1691 as a plant. It's very, very similar to the one I actually took pictures of last weekend in the same river. So they're persistent little beasts. They are, of course, not plants. They're animals. They're true sponges in the phylum Porifera, and each one is a meta-individual. It's a colony of individual cells that work together. There's no true tissues, there are no organs, but the structure it's a filtration system. It has pores, and water is drawn in the pores and out through other pores, and they're gathering food from the water column. To do that, they have specialist cells. The cells specialize depending on where they are in the structure. There are cells that specialize in having flagellae, which pull the water through. There are cells that specialize in phagocytosis to engulf particles, particularly bacteria, which are a key part of their nutrition. There are cells that excrete structural devices. The structure is often really made of silica spicules, which give it that solidity. That's why it can stand up. So there's quite a lot of things going on, considering that they are all the same cells. So there's this internal cavity within the sponge, and pores that pull water in and pores that push water out. There's collagen fibers. There's the silicon spicules, silicaceous spicules, and they filter water. There's some research that says a finger-sized sponge can actually filter 125 liters of water a day. So there's a lot of water passing through these things. And so a big sponge, or even a colony of a lot of small sponges, is doing a lot of stuff to the water column. It's pulling that water through, and it's taking out particles and bacteria, etc. So it's doing a lot of things. Their life cycle, just very quickly, they are obviously sessile. They do not move. They grow from a particular place in the spring. They usually die down in the winter. They survive the winter and in dry conditions with gemmules or gemmules, depending on how you want to say it, which are propagules of sponge tissue enclosed in a hard structure. And they produce those in quantity in late summer. So about this time of year onwards. And those gemmules are actually quite important in identification. We'll come back to those a bit later. Sexual reproduction they do as well. They have oocytes, which remain within the maternal structure, and spermatozoa, which leave the structure and go to another sponge to fertilize an oocyte. And then when it's fertilized, planktonic larvae, this is the only time they actually swim about, goes somewhere, can swim off somewhere else and establish a colony. The gemmules will also establish new colonies, but that's asexual reproduction. And so a lot of reproduction will probably be either gemmules. In terms of worldwide sponges, there's 8,000 odd species of porifera, but only 250 of them in water, fresh water, and the order called the Spongilidae. All the British species are in the family Spongilidae, and within that family we have 6 species, these 6 listed here. They all vary in appearance. They all tend to be found on hard substrates, and they all at various times look like each other to some extent. So it's difficult to identify them. These next few slides have got a lot of words about each species, and I haven't got time this evening to go through all of it, but basically Spongilla lacustris, the one we were looking at first, is the strongly branched one with the big fingers. It can be massive, up to 1 meter, although it can be tiny. It will be green in light and yellow or grey in shade. Similarly, Ephidacia flaviatilis is also green in light or yellow or grey in shade, and that can also have little proto bumps, so that can grow into little bits that stick up as well. But all the rest are more encrusting, more lichen-like, but these top 2 can also be lichen-like. So just looking at them is difficult to tell them apart. Just going through those quick, well, I think it's on the next slide, their distribution, I think I've got on the next slide, well, their habitats. The Spongilla lacustris is basically everywhere. And it likes to live on rocks, structural masonry, tree roots, also on bivalves, which is interesting. Several of them will grow on bivalves, so you can find swan mussels or even zebra mussels. I mean, a bigger mass of zebra mussels can often have sponges growing on it, and could even control their spread to a certain extent. Ephidacia flaviatilis is rivers and streams largely, but it also grows in lakes and ponds. Again, hard surfaces. Ephidacia moellii, rivers and canals. Eunapius fragilis, rivers and lakes. Rassicula ryderi is lakes and rivers. And then last but not least, Trochospongilla horrida, which Karen and her PhD found as a new species to Britain, has only been found so far, as far as I'm aware, in the Exeter Ship Canal. That was by Karen a few years ago. Distribution. This is one of the key messages I want to put out tonight, is the distribution of sponges on formal records. National Biodiversity Network records is what we're seeing in this image here. This is the 2023 map from the NBN. And basically, it suggests that they're only locally common in a few places. It's a load of baloney. They occur virtually everywhere. So why aren't they on the NBN maps? They aren't on the NBN maps because hardly anyone records them. I mean, it's just as bad as anyone else. I've got a load of tubes upstairs of sponges in which I haven't sent off the records of. But I probably collect them more than most people do, and most people don't collect them at all. So there's this massive gap in records, partly because they're difficult to identify, but also often because they're difficult to find. The encrusting ones, you tend to overlook. You just don't see them because they're encrusting an underwater structure, and you're not looking at underwater structures. Karen's work, which is on the top map there, the map of Britain, the distribution map she's got there, you could see the lines. A lot of her sites were northwestern canals. You could see the line of the canals on that map. You could see that wherever she went on canals, she found them. Similarly, the lower map, which is Southwest Ireland, that's the recent work done in Ireland, where they were looking at rivers and streams, and you can see the lines of the rivers and streams. In other words, everywhere they looked on the rivers and streams, they found sponges. So they are actually everywhere if you look for them, like these two studies did, but most people don't look for them. And if you look for them, it's difficult to identify them because, as I've already hinted, neither form, neither the shape of it, nor indeed the habitat are reliable diagnostics. You really need microscopic examination of the spicules, which are diagnostic. So the shape and the size of the spicules are distinctive for each species. And so that's the way to tell them apart definitively. There's 3 different types of spicule, which we can't go into great detail tonight, but there's the megascleres, the microscleres, and importantly in the gemmules, the gemmular which are very distinctive per species. And so it's quite important that when you are sampling sponges, if you can do it in late summer, early autumn, when the gemmules are about, you're going to get more chance of a firm identification. The image on the left is showing you what some of these spicules look like. A lot of them do look very, very similar to each other. That's another of the problems. Why you want the gemmules, because you need the more different. The gemmule ones are more significantly different to each other. You can look at these down a microscope, and you need a compound microscope, but you do need to clean them of the sponge tissue. We'll come back to that a little bit in a second. So this is a few of the types of spicule you find, and you'll see that most of them, the long thin ones, are the megascleres of different species, and they do look worryingly like each other. But the wacky ones, the odd-shaped ones, are the ones from the gemmules, and you can see that they can be quite species distinctive. So this is why gemmules are so important in identification. This slide just shows you a little bit about what those spicules can be like per species. But if you look at Spongula lacustris versus Ephidacia flaviatilis, the megascleres of both look surprisingly similar, don't they? They're 200 to 400 mu long, 200 to 450 mu long. They're needle shaped. Yeah, well, yeah. So you really do need those gemmules, because that's where they start to differ. If you look down the whole list there, the megascleres are quite similar in most of them. Now, Karen, I hope you forgive me for reformatting your key, which was originally in your paper a diagram, but I've written it out here as a key, the format that a normal key from an ID book, but it is the same key. So this is Karen's key, and it's showing you how you can differentiate these species just on the spicules. But to do this, first of all, you need your spicules. And compound microscopes are essential, obviously, and the specimen needs to be prepared. If you just stick a lump of sponge down a microscope and put it under magnify it 400 times, you get an image like this. which you could see loads and loads and loads of spicules in there, but there's no way you're going to start working out how big they are and what shape they are, etc. You need to clean that tissue off. And in a laboratory condition, you would clean it with nitric acid to dissolve the organic material from the sponge and just leave the spicules. But most people haven't got a laboratory with nitric acid. So they are more difficult to do in a domestic situation. It can be done subtly with a bit of bleach. This is just an example of an old Russian microscope I've got upstairs that shows that you can isolate spicules quite with determination. You can get them. You need to measure. If you want to measure them, you need, of course, your graticule in your eyepiece, and you need your stage micrometer down at the bottom as well to whatever the word is. Calibrate, that's the word I'm looking for. Calibrate it. So you can do that, but it is quite a challenge. But I would really say to people, give it a go. It's an interesting thing to do. Changing the subject completely, having wound you up to how difficult they are to identify, this is where I want to go talk about the associated species. The size of them, as I've said earlier, means that they are almost certainly going to be eaten by something, and they are indeed eaten by some very specialist invertebrates. The sponge flies, the Ciceridae, which is a worldwide family of lacewings, which has evolved to grow in the worldwide group of freshwater sponges. They occur all over the world, the Ciceridae. Also, some caddis, some leptocerid caddis, and a few from other caddis families will eat sponges, but there's a few in the Ceraclea genus that are sponge dependent. They only eat sponges, or they need them within their life cycle. They also use them in their cases. We'll come on to those in a minute, plus a few chironomids and a few other bits and pieces. So sponge flies, they're the most remarkable of the lot in that they only feed on sponges. They're named after sponges. They live for sponges. That's what they're there for. They are lacewings. We have 3 species in Britain. They're all in the Cicera genus: Cicera nigra, Cicera dalei, and terminalis. The adults are dark lacewings. The larvae are these cute little things. They're only about a centimetre long, even when fully grown, which feed on sponges. They have this interesting life cycle where the adults lay eggs which drop into the water. The larvae then have to find a sponge. They live and feed in that sponge through 3 instars before they emerge and overwinter on land as pupae. And the larvae will be outside the sponge, feeding on the sponge, and they feed with specialist tubular mouthparts, which they put into the sponge tissue. So that's sponge flies. They're really rather weird things. And this is where it gets interesting in terms of records. This is sponge fly distribution. This is a map I produced for the 2023 review of the 3 species of sponge fly and their records in Britain. And as you could see quite quickly, there are far more records of sponge flies than there are of sponges, which is obviously impossible, because all of these sponge flies must have been brought up and lived as larvae on sponges. And you compare that to that map that's just appeared in the right-hand corner there, the map of sponges, and you think, well, there's something very wrong with that map of sponges if the sponge flies are all over the place. So they are very under-recorded. Now, moving on to the other big sponge eaters, the sponge-eating caddis. We have 4 species of those: Ceraclea nigra nervosa, fulva, albimacula, and senilis. Senilis, Ian and I have been talking a bit about this evening, actually. That we think they're probably all obligate feeders. I had senilis down as a facultative feeder, one that also eats other things and possibly could survive without sponges, but we think it probably is obligate because we can't think of where I found any record of it eating something else. So that's probably all obligate. They feed on sponges in certainly the later instars. Some of them in their early instars feed on other detritus, but basically they're very similar to the sponge flies. They rely on sponges. They take it a step further in that they make their cases out of sponges. There's also a few others, the Ecnomus tenellus, which Ian was saying earlier to me, it's that the records of that appear to be more numerous where sponges exist. So that looks like it does rely quite heavily on sponges. Some other limnephalids and some hydrocycae also use sponges in their cases. And so we've got this, the life cycle of these is fairly normal. They have instars, they overwinter as early instars, the adults lay the eggs, etc. So they are very similar in many ways to the sponge flies, but they do have this oddity that they have cases made of sponge. And the cases, unlike most caddis cases, the sponge is still alive. So the sponge on the caddis case is going to continue to be filtering water on the caddis case. It might even carry on growing, and if when the caddis moves off and emerges from the water to pupate, then it may well have created a new sponge colony where the case has been discarded. It's quite an interesting bit of distribution there because caddis are moving the sponges about. If you look at their distribution, you get exactly the same picture, but even more definitive than the sponge flies. These 4 species of caddis occur all over the country, and yet they need sponges. And therefore, sponges must be all over the country, even though the official map says they aren't. So it is really demonstrating that everywhere you get these species, there are sponges, and yet we the ecologists, the species recorders, we don't seem to be recording them. There are a few others. There's some British species of chironomid. There's a couple of chironomids in Europe as well that are sponge specialists. Also, some of the water mites seem to be almost certainly sponge specialists. It's not quite definitive. Some bigger invertebrates, the amphipods, particularly the introduced Carophium species, also seem to eat sponges. And a curiosity down the bottom there, the sponge flies are often described as parasites of sponges, but sponge flies also have their parasites of their own, because there are some pteromalid wasps that colonize the pupae of the sponge flies. So there's a whole long list of things that have their origins in sponges. And very briefly, just thinking about eating sponges, bearing in mind that a lot of those things are very small. The Sisira larvae, the sponge fly larvae, are only about 10 millimeters long, and yet these spicules, some of these spicules are nearly 0.5 millimeter long themselves, 400, 450 mu, that is. So you've got a body that's 10 mil long, and you're trying to eat something with siliceous spicules that are 0.5 millimeter long. It's going to do some funny things to your gut. And so looking into that, it's quite interesting to see how the groups have done it. Sisira, of course, when you think about it, doesn't eat the spicules at all. It's got sucking mouth parts. So it only takes the sponge tissue. It doesn't take the spicules. The caddis, it's not quite clear what's going on there, but studies of vulva suggest that it has a thicker gut lining, which may be protecting it from these rather sharp-looking spicules. And a study of one of the chironomids showed that the spicules seem to have lost their points. Somehow it had shaved the point off before it swallowed it. There's some interesting things going on there. But that only applies to these tiny things. There are larger sponge feeders, which can include signal crayfish, for instance, will eat sponges. And of course, they've got a much bigger gut, so a few spicules probably don't worry them at all. That's a bit of an aside. Anyway, I want to move on quickly now to finding sponges, because as well as identification, to study sponges, first of all, you've got to find them, which can be a bit difficult. Again, Karen might have some thoughts on this at the end, because she did a PhD, which basically was based on finding sponges. But this is the River Thames in the picture. This was last weekend. This is in the middle of Reading. This is Reading Bridge, and there's this curious little walkway underneath it, which creates a little backwater on one side, and which is known for its sponge populations. There have been records from there before. And last weekend, I went to have a look and to try and photograph some underwater and possibly get a bit of video. So we went down there, my better half and I, and we went down onto this walkway, and we were quite disappointed at first sight, because I had promised lots of sponges immediately visible, and Caroline, I think, wasn't convinced they were going to be there when we looked at this. The water was you could see down about ½ a meter, but you couldn't see any sponges. But I did have a camera on a pole, a GoPro camera on a pole, which you have to use blind underwater. I don't know if you ever used one of these, but they connect to your phone with an app, but only when they're in air. If the camera goes underwater, it loses the link. So by the time you stuck the camera in the water, unless you're actually diving down with it, you can't see what it's filming. But what's interesting here, and this is what I wanted to stress, is that even an unpromising looking bit of water like this, you stick your camera down there and you'll see what I mean. And the next slide is a 60-second film of me dropping the camera in the water. Well, not dropping it in, I'm holding it on the end of a long pole. So here it is, it's going down into the water, and you can't see anything. And then all of a sudden, there is a wonderful example of a branched sponge. This was the fourth take, so I knew roughly where it was by this time. So this is how I got so much of it. But you can see there's a few roach swimming around as well in there. But you bear in mind, I can't see what I'm filming, so I'm thinking, oh, have I got the best bit yet? And just as I do get the best bit, in a few seconds' time, I get a nice picture of it growing out the substrate. Yes, that's a really good shot, but that's the point where I decide to bring it out of the water. And you just get a picture of my head at the end. So I'm putting that in just to make the point that even an unpromising looking place will have sponges. The reason I went there particularly, by the way, was that these pictures on the left here were taken by Victor Desiulis in 2011, when he found some quite spectacular sponges just there. Probably Ephidaceous species, although the one I just got that little video of is probably Spongilla. So it does pay to go back to sites to see what's going on, and it pays to have tools with you. In the case of last weekend, I had the camera on the pole. It would have been quite useful to have had a grapnel as well to get some samples, because it was deep in that water and I couldn't actually get a specimen. All I could do was film it. Finding sponges, and a second example, this is a canal bridge on the Grand Western Canal in Somerset. And I was virtually certain there'd be sponges here, but you look into the water and you can't see anything. You could scrape the walls with something, but I like to actually see them. But eventually I realized where they were. The coping stones just under the bridge stick out over the water slightly. And so there's an overhang, and there's a whole line a wonderful line of sponges growing underneath that overhang. You can see them in these pictures here, which are taken by leaning out with a phone camera and just with a, on the selfie mode, so you can see what you're taking a picture of, and you can see this line of sponges all the way along here, which are some form of one of the Ephidaceous species. So very, very distinctive place to find them, but also impossible to spot unless you deliberately went looking for them. You would just not see these if you were doing an ecological survey. You wouldn't even sample them if you were doing invertebrate work with a net. You just wouldn't get this. You've got to actually look. Another example, this is a more extreme example, and I don't recommend trying this very often. This is the Caledonian Canal in Scotland. It's a big canal. And this is a lock that's been drained down for maintenance work. And on the walls, in the lower sections, you can see what look like big lichens on the side, which are sponges, probably Ephidaceae. And you would never know they were there. It's virtually impossible to sample those unless you're in this circumstance that you're there on an engineering inspection with your hard hat on, and you can go and get one, scrape one off the wall. But you would never normally know they were there. Another good place to look, which is again on canals, particularly on the offside, that sponges will form a glove over exposed tree roots, particularly older roots that are growing into the water. Probably again, almost certainly Spongilla lacustris, and you can actually put your hand in and lift these out. Quite spectacular growths of sponge. And then going from one extreme to another, this is last weekend on another river. I've arrowed them in the picture because they're so obscure. My foot's for scale there. We're looking at two tiny little sponges. It's the same size as I had earlier with the exposed ones out of water, but these ones are underwater. And again, they look like a little bit of moss, but they're not moss. They're sponges, and very easy to overlook. Very easy to not find. These are conspicuous because they're green. Just think how difficult it is to spot one that isn't green. So some of the others, the Spongilla and Ephidaceae flaviatilis are reliably green in light. The other species tend to be less green, and so they are difficult to spot. A little bit about finding the sponge feeders, if that's your thing, rather than the sponges. If you do sample a sponge and you sample it a decent specimen, so I've got there not a hasty scraping, don't just scrape a bit off with your fingernail. That's enough for ID perhaps, but it's not enough to get caddisflies and chironomids and sponge flies. You need a bigger specimen for that, and you'll quite often find that they're in there and they will emerge, or you can dissect them out and they'll be a bit annoyed that they've lost their home, but you'll be happy that you've got the records. And of course, those species all live elsewhere when they're not in the sponge. And last bullet point there is an interesting one as well, that and someone wrote to me about this the other day, that they found some sponge flies, so now they know where to look for sponges, which is an interesting way of looking at it. If you found your sponge fly, a swarm of sponge flies in the evening by the side of your river, pond, canal, whatever, you know there are sponges there, so you should be going to look for the sponges soon or next year or whatever. And what to do with sponges? Well, you really need to be collecting them in late summer when the gemmules and the spicules and the gemmules are available. You really need to put them into alcohol. A lot of people say, well, I haven't got any alcohol. You really need 70% ethanol or even 100% ethanol. But you can get HMSRC do issue licenses on an industrial denatured alcohol, so you can get supplies. Or as I think it was Karen who said this to me the other day, you could just use vodka if that's, or you can drink it, but you can use it for specimens. And then you can examine them later yourself, or try and find someone who is more expert to look at them for you. But if you're doing it yourself, you do need the microscope and the cleansing and the determination. But it's worthwhile doing. The associated species, this is we can talk a bit about more of these in the Q&A afterwards, but there's a lot you could do with those. Again, the recording schemes, there are recording schemes for those, and there are previous Tanyptera Trust webinars on them. Ian did a couple on caddisflies, so there's one already available on the web about caddisflies and caddisfly adults. And spongefly identification. Adult spongeflies are covered by James Jepson in his webinar on lacewings. That's the lacewings part 2 webinar, not the lacewings part 1 webinar. So there's a lot of information on those. And as mentioned earlier, Ian Wallace is here today, so he can take any questions about Trichoptera and indeed probably on spongeflies. And to mention Karen again, Karen is here as well, so she might be able to answer any questions about, and even put me right on some things possibly, on sponges. So concluding remarks before I move on to some references at the end, just to say sponges are probably very common, and yet the national distribution map says that they aren't. But that is completely shown to be utter nonsense by the maps of the spongeflies and the sponge-dependent caddis. We could do a lot more work on them. We could do a lot with distribution. We could do a lot with biology. We could do a lot with the insects associates. But the recording of them is challenging, both the collecting and the identifying. There's no recording scheme. It would be lovely if there was one, but it would be quite difficult to set up because it would need some way of some system of making sure they were determined properly, which would really require someone to be probably being paid to take the specimens, and that doesn't exist at the moment. But anyone can give it a go, a microscope and, as I put here, determination. So it's worth having a go. And with that in mind, these bits of reference would be useful to you. There's my little review I did in 2023. There's Karen's work, which was written up in 2019, her paper in invertebrate zoology. And then there's John and Letitia's work in Ireland, which was written up in 2014. And if people need copies of those, I can email them by whatever means is appropriate to the Tanyptera Trust to send, or you can write to me at my email, which is on the screen there. I think that's all I've got to say for the moment. So let's move on to questions.
Okay, thank you, Jonathan. That was absolutely fascinating. I have to admit, I didn't know a lot about freshwater sponges before today, but yeah, I really enjoyed that, and yeah, particularly learning about the sponge eaters. Yeah, I never knew that caddis used sponge in their cases, so yeah, really fascinating. So yeah, if you want to stop sharing your screen, then we'll take questions.
Yeah,
we've got a couple in the chat. The first one is from Caroline. She said, it sounds like if it's a large fingered sponge, then it's Spongilla lacustris. Is this correct and okay for an identification, or do you really need the microscope ID as well to confirm it?
I would say it's probably correct. Karen might have a view on that, but I think if it's a significantly branched large sponge, Spongilla lacustris. What do you think, Karen?
I agree.
Good. Thank you.
Okay, thank you. So we've got another question from Lilia, who says, do pollution levels affect sponge growth?
It depends on what sort of pollution. They are quite tolerant of a lot of different water qualities. and they do filter a lot of stuff, so they will be affected by pollution. But it depends on what that pollution is. They will survive in quite turbid waters. They'll survive in quite bacterially rich waters. So for bacterial pollution, for instance, downstream of sewage works, they will, in some respects, be quite happy because there's all those bacteria to phagocytose, or whatever the word is. So they are quite tolerant, but it depends on the type of pollution.
Okay, thank you. We've got another question from Dave Bolden, who says, how much sponge does one need to take to identify it?
Not a lot. Only a sort of a centimetre square would be fine, more than ample, to be honest, because you'll end up with masses of spicules. But what you really do need, particularly to be absolutely sure, is the gemmules. So the gemmules or gemmules. So look for those at this time. Yeah, I've realised after I started talking about gemmules just now in the talk that I hadn't got any pictures of them in there, which is a bit stupid, but they are visible to the naked eye quite often, the gemmules. So, and they're small little particulate things that around the base of the sponge, which will appear at this time of year. So if you can collect some of those, then that's better. Again, Karen would have a view. She's probably sampled more sponges than anyone.
Yes, I've just tried to put a picture of some gemmules in the chat, and yes, you don't need much sponge at all. I think a centimetre, square is fine.
Okay. And yeah, thank you, Karen.
Thank you. Caroline asks, I think you mentioned they can grow on zebra mussels and may control spread. Is that right? And if so, how?
Well, when they grow in sufficiently large masses, they'll stop the mussels from opening. So they can effectively kill. Zebra mussels are like marine mussels. They connect together on byssal threads. And are a problem, of course, in so many places because they obstruct pipework, etc., and stop gates closing. And so a large mass of sponge on a mass of zebra mussels can actually kill off the zebra mussels. And there have been some records of this happening. However, you've got to counter that partly with the fact that a massive growth of sponge can be a problem in itself. And so a massive growth of sponge, while the zebra mussels might have been a problem, it growing in a mass of their byssal threads in your pipe, the intake to your whatever it was. If they're killed off by a massive sponge, that's also going to be blocking your intake to your whatever it is. So sponges can control zebra mussels, but they can also be a problem themselves. And I doubt they are let's get this into context. They're only controlling zebra mussels in very local circumstances. They're not going to be a solution to zebra mussels.
Okay. Jez Martin asks, is there scope for new species to be discovered?
Oh, I think undoubtedly. Karen found a new one to Britain just by looking, and that was that added one new one to. We only had 6. We only had 5, and now we've got 6. If you can do that in 3 years or so of a PhD, then a bit more intensive study, you could probably find a few more. So there almost certainly are more about, and I wouldn't be surprised if we're going to get some new ones coming in through the usual how do new ones arrive channels, with aquaria waste being chucked into water bodies. So almost certainly there could be new species out there. Although, to be honest, we have enough challenge identifying the 6 we've got.
Okay, we got a couple more questions in the chat. So from Laura Mulholland, she says, "Hi, Jonathan. Really interesting talk. Thank you. I'm an ecologist at the Canal and River Trust, and I often see sponges on the lock gates when we lift them out of the water to change for the new gates. It feels unethical to lift them out of the water, essentially killing the colonies. Would there be any way of trying to preserve them and chuck them back in?"
I don't know. It's they basically, you've got to remember they're unicellular animals that just live in a colony. So they're not like plants, which if you uprooted them from the ground and left them and chuck them back in, they might just die. They can recolonize structures. So you could put them back again. You could. Now, the only time I've ever done this successfully, or known I've done it successfully, was way back in the 1980s when I worked on the Montgomery Canal, when we took a sponge from the canal and put it in our office aquarium. And we were doing a lot of work on that canal at the time. We put everything in that office aquarium. And I'd read up that sponges were very difficult to keep alive in an aquarium, so I didn't think it would live, but it did. It started growing up the side of the aquarium, and that was when I saw my first sponge fly larvae. It was that on that sponge, looking through the glass of the aquarium, I saw these larvae living on the sponge. I thought, good grief, one of those. Now, that does tend to suggest that you can transplant them, not to an aquarium necessarily, because the problem with an aquarium is that because they're filtering all of this water all the time, they need a constant supply of nutrient-rich water, and in an aquarium they're going to run out of that quite quickly. That particular aquarium, we used to put new samples in it virtually every day, so we were probably feeding these sponges stuff. But yes, I can't see why they shouldn't try and be conserved. But you've also got to bear in mind that they are ephemeral. They only that growth you're seeing is only one season's growth, and so it will have propagules, and so it will probably just grow again, even if you just chuck it in the canal and that whole growth disintegrates and dies. A little bit of it will probably still be alive and may well survive till next year. So it's a tricky one, but I suspect it's worth doing.
Okay, interesting. Thank you. So Lilia asks, do you think their water filtering capabilities at all helps to keep water bodies clean?
It does to an extent. I mean, one of the interesting things about sponges, and there were some studies of this in Northern Ireland a few years ago, is their ability to accumulate material from the water, particularly bacterial material. And so there was a study done below sewage works specifically to look at how the sponges were at least could be used as an indicator of the fecal coliform, etc. pollution, because the sponges were filtering the water constantly, and they were particularly interested in the sponge, if it's thinking at all. The sponge is wanting bacteria. But if you take samples of sponges downstream of the sewage works, you will find, in theory at least, a record of the pollution of that sewage works and how it decreases downstream, because you've got a record within the sponge structure of the bacteria that are being caught. So they are filtering, in that sense, they are filtering effectively, in that they're taking stuff out. Studies of them in the tropics have suggested that they are having a significant effect on water quality where they occur in large masses. Whether any of ours occur in sufficiently large masses, I couldn't say, but they definitely have an effect. How significant that is, I don't know.
Okay, following on from that, Lilia asks, are there any conditions where they wouldn't grow at all?
Well, highly polluted situations, also anywhere with no hard substrate. So something that was just a silty bed, they do want something solid to live on. Because they need to start from somewhere, they need to start from a solid surface. So anywhere that's has no solid substrate, I often think of them. That's this. You go for this way. Madness lies when you start thinking about what did things do before man, because when you look for sponges, the places you find them most are on man-made structures. And so the softer pre-man-made waterway might have had fewer sponges. It's similar arguments to what did house martins do before houses, or barn swallows do before barns. So there must be circumstances where they would not grow well, but heavily polluted areas. But I think they probably occur in most environments if they are there.
Okay. And Bill asks, could artificial substrates be used to find and monitor sponges?
Almost certainly, I think, yes. Again, you think of the canal type situation, the lock gates, the bridge holes, and indeed there's some work done I referenced it in the British wildlife paper done on the Mekong River, so a long way away, but they were studying sponges on rafts, and looking at how that was affecting the local biodiversity, and finding that rafts with sponges were more diverse areas of the river than the rafts without sponges. And I can't remember off the top of my head, but I think those rafts were put in deliberately for the sponges. So yes, I almost certainly you put you stick a timber, preferably one that's not been tannalized too much, stick a timber in some water, and you probably get sponges colonizing it at some point, or just a rock in some water. It's a matter of exactly where you do it. They don't seem to tolerate, the big ones don't seem to tolerate too much disturbance. So if you want a big fingered sponge, you want it to be in some side water where it's not going to get knocked about. So if that's what you were encouraging, then find a side water and then start putting a few rocks in it and see what happens.
Okay. Dave Baldwin asks, do some prefer alkaline substrate and others otherwise?
Oh, yes. Well, I think I think the jury is largely out on that. I mean, Karen again might have a view. Water quality, you found them in quite a wide range, didn't you?
Yes, I don't think the data is there to say that, you know, what the limiting factors are. I think some species are very tolerant of changes in pH.
Yes, so they're quite Catholic, really. But as you say, there probably isn't enough data, really.
Correct.
Thank you.
Okay, thank you. Yeah, we've still got another question in the chat. Everyone's really engaged, so the questions keep coming. So Bill Brearley asks, do sponges feed on microscopic algae as well as bacteria?
Good question. I imagine they probably do. I don't imagine they're that fussy, to be honest. As long as it's if it's a small planktonic cell that happens to pass one of their phagocyte cells, then it will just scoop it in and eat it. I don't think they're particularly fussy about what they engulf.
Okay, yeah, that's the end of the questions in the chat, unless anyone wants to ask any more. One more. Do sponges occur in fast-flowing streams in Highland?
Probably. again, no one seems to look for them. The Irish study was looked at some quite fast-flowing streams, and that's worth looking at. The problem in a fast-flowing stream is actually finding them, because they'll be on rocks, but they may be under the rocks, and so you do that dreadful thing when you're looking for, you did when you were a child looking for freshwater shrimps. You stand in the stream, you start turning every rock over, desperate to find something that might be a sponge, and quite often you don't find one at all, or you can't identify one as a sponge. So I think they probably are there, but maybe not at very high altitudes, though.
Okay. Yeah, I think that's all the questions now. Did you have anything to add, Ian?
Yes. Can I clarify over the sponges on cases, and Jonathan?
Yeah.
All caddisfly larvae make a case. The inner part of the case is made out of silk, but well, it's a flexible material. In the case of some of the sponge feeders, it seems they almost encourage the sponge to sort of grow into this structure. So the sponge becomes an integral part of the case. But there basically is just a thin, rather flexible, horny material, which is where they're living in. The exception is Serratia nigronervosa, which has to overwinter all the way from about August or September through to about May. And that makes quite a tough case. It's thick, it's reinforced with sand grains. The other thing I'd like to say is, you did mention isopropyl alcohol is the material that I use now for almost all the preservation, and it's very cheap, and you don't need any licenses or anything
Yes.
else for that. And the final thing is, which it's one of the intriguing things, and it could be water quality improvements, but one of the sponge feeders, Serratia senalis, has increased its distribution. In the 1950s and 60s, it had struggled to reach Leicester. It is now in North Lancashire and in Yorkshire. I suspect it has liked the canals. I don't know if the water quality has improved in the canals, but it certainly likes the canals. And I think it may have probably reached the northern end of its distribution simply because it's going to have problems. We run out of canals.
Yes. Yeah. You want to get to the northern end of the Lancaster. It's a big jump to get to the Forth and Clyde.
I'm not sure if it's yet in the Lancaster Canal, but certainly it's a very fine insect.
Interesting, because yes, an interesting point, because the canal water quality has improved, although it's also, there's a lot of problems with eutrophication and basically storm sewer overflows, etc., which has got worse. But in general terms, it's improved. It would be interesting if it's in the Lancaster, though. I mean, the northern reaches of the Lancaster must be, it would be worth going to look for it if it's not been recorded, because that's the northern reaches, well, after the M6 crosses it. It's unnavigable from there up to the Killington Reservoir, and that's a really good section for biodiversity.
Because it obviously flies quite well, because having reached the Lancaster, then it's only a short flap and it's in the Lake District. Which should be good. Fine. I have a sponge-related question here, which is, I was interested to see how well they could tolerate silty water. The reason that I asked that is because on the marine sponges, in the Mersey estuary in particular, it is so silty, and on the Dee as well, that you don't get sponges normally growing on the rocks. They come at the top of little clefts where there isn't so much silt getting there. So it obviously, I was intrigued that they can, survive quite a bit of siltation.
Yes, well, that first picture I showed of the white fingered sponge, that was that looked like it was growing through silt, and I suspect it started life before that silt was there, and it's just carried on growing through it. But silt obviously does affect them to some extent, but it's an interesting point about canals in particular, in that those big growths I was showing earlier were in disused canals, and disused canals don't tend to be very turbid. They have silt, but it's not often disturbed enough to coat things. But in used canals, the silt is disturbed all the time by boat traffic, and so it's continually being churned up and depositing on every surface there is. So everything under the water is constantly being covered in silt all through the summer months. The worst possible time to be covered in silt if you're a plant or an animal. But even in those, we get the encrusting sponges still. But there's interesting points to think about there, that the encrusting ones quite often in canals are on the vertical surfaces, and silt will be falling off those. So it may not affect them that much. It's a curious thing. It could. There's another couple of PhDs there for someone, I think. But
in the Shropshire Union Canal, I found that it's often at the narrows, the bridges, which is where you often get the sponge, and whether it's the extra water flow is giving more food for the sponges, or whether it's, as you've just suggested, possibly it helps to keep those areas free of silt. Yeah, interesting.
Okay, so if there's no more questions, I think we'll leave it there. Yeah, so thank you, Jonathan, for a fantastic talk and for answering all those questions so thoroughly. Yeah, and thank you to everyone else for joining us.